Thanks, Stephen. So I run a research group in the University of Cambridge in the Department of Pharmacology, and my lab, among other things, works on heart regeneration. So today I was just going to show you some of the basics around what we're trying to do to develop new methods to regenerate the heart so that we don't have to transplant patients. So Stephen's gone over this beautifully. Heart transplantation works and is transformative to patients. However, there's over 100,000 heart failure diagnoses each year in the UK, and only 200 transplants take place. So it's, there's a really huge unmet need. And so we really need to work on alternative solutions for heart regeneration. Okay, so I asked you the question, why don't human hearts heal? So, if you damage your skin or you break a bone, that can regenerate fine. But why doesn't the human heart do this? And for this, you need to kind of zoom in and look at the cells that make up the heart. And the cells made up of these muscle cells or these cardiomyocytes. They're just so beautiful when you look down the microscope at them. They have these beautiful stripes. They're called sarcomeres, and they help the cells beat and pump the blood around your body. So the problem with these is that after a myocardial infarction, up to 1 billion cardiomyocytes can die within the first few hours following an infarction. And these cardiomyocytes, they can't make new cardiomyocytes. So in your skin cells will proliferate, make new cardiomyocytes and replace the old. Cardiomyocytes don't do this in a human or in a mammalian setting. So what happens is you go from this beautiful, healthy-looking heart to a damaged heart. And so you've lost cardiomyocytes, you've lost that pumping ability, and your heart goes through this pathological remodeling where it changes shape to compensate. So there's a few things we can do, and this is a slightly busy slide, but it's essentially there's really 2 avenues we can take. So what we want to do if we want to regenerate the heart is to remuscularize it. We want to replace back those cardiomyocytes so that it can beat again. And so there's cell-based therapies, and this is what Professor Sanjay Singh works on, and I'll briefly cover this. So either you can directly inject them into hearts, or you can make an engineered patch and kind of attach it to the heart, or what my lab work on is this cell-free type therapy, where we try and give them, and this is just pictures of different ways of delivering drugs or therapies, we try and stimulate the cardiomyocytes to generate new cardiomyocytes. So you endogenously regenerate your own heart instead of adding them back in. So if we talk about cell-based therapies first, and this is what Professor Singer worked on, essentially it's making cardiomyocytes, injecting them into the heart and remuscularizing. But where do these cardiomyocytes come from? And these cardiomyocytes come from embryonic stem cells or stem cells. And stem cells are these amazing cells that can self-renew and keep making new cells. And what you can do with them is you can give them various different growth factors, and they can make every single cell in the body. And so you can stimulate these stem cells to make any type of cell. And many labs across the world do this, looking at regeneration and various different developmental biology. So this is what my lab and what Sanjay's lab do. We give the cells, these embryonic stem cells, these different growth factors, and we generate cardiomyocytes. This is from one of my students over the 10-day time course. And what's really amazing, and normally when you look at cells down a microscope, they don't move. They just sit very statically like this. And they're beautiful and just wonderful to look at. But cardiomyocytes, they beat in culture, and it's just the most fantastic thing. It never gets old. I love going into the lab and seeing beating cardiomyocytes. So this is what we can generate. And then what various researchers, including Sanjay, have been doing has been injecting these back into hearts. And here we've got hearts. So in the red, it's labeling the cardiomyocytes, and in the blue, it's labeling the scar tissue. And this is after an infarct, so there's lots of scar tissue in this heart. And in this heart, they've injected human stem cell-derived cardiomyocytes, and they've been labeled in green here. And you can see that they're really remuscularizing this heart, and it was able to gain extra function. However, there's so much work to do. So there's a number of issues with this kind of therapy. only less than 10% of these cardiomyocytes survive. They don't necessarily engraft with the host very well. They don't necessarily regain electrical connectivity. There's problems with immunogenicity, and so the immune system can try and remove them. And so there's a lot of research going on in this area, and this is what Sanjay's lab works on. So if we look at the other type of therapy, and this is what my lab work on, this is a cell-free therapy, and this is really trying to stimulate the cardiomyocytes within the heart to generate new cardiomyocytes. So you're making your own new cardiomyocytes rather than injecting them in. So you'd have a heart, and if it got damaged, you'd have loss of these cells. Then we'd inject this therapy, and the cells would proliferate, and you would get new cardiomyocytes. So how do we know it's possible to stimulate cardiomyocytes? So, they don't normally proliferate, and they're very difficult to make them proliferate. So how do we know that we can do this, and this is actually a method that we can use? And for this, we look at other experimental models, and the fish and the axolotl or the newt are really regenerative models. You could damage their digits, their fins, their hearts, and they'll just completely regenerate them. They're just the most amazing species to study when you're looking at regeneration. And this is just some pictures from Ken Poss's lab. Here at the bottom here, we've got a damaged fish heart, and after 60 days, completely regenerates. And it does this by cardiomyocyte proliferation. So the cells are making new cells. But again, you ask the question, well, these species are different. How does a fish relate to a mammal? And actually, if you look at juvenile or one-day-old mouse pups, and you do exactly the same thing on them, and you damage them, and they cause a damaged region, they can regenerate as well. So this is actually possible in a mammalian setting as well. So we know it's possible to stimulate mammalian cardiomyocytes to proliferate and regenerate a heart. So what factors do this? Many labs have been exploring various different factors to try and stimulate this proliferation. And my lab work on a protein called MYC, and it does many things, but its main role is to drive proliferation. So we thought, great, let's put this into hearts and see whether we can proliferate these hearts. And when we added it in, and we did this in a kind of genetic factor way, so we weren't adding a drug, we were just stimulating the heart to overexpress this protein, didn't do anything. So we were like, oh, what's happening? And we spent a long time looking for other factors to try and help drive this proliferation. We found this other factor called cyclin T1, and when we overexpressed them together, we could drive proliferation in the heart. And this picture here just doesn't look very nice here. But I'm going to show you this here, the green is the proliferating cardiomyocytes, and this is where we've overexpressed these two factors, MYC and cyclin T1. And I hope you can see from the back there that we've got a lot of green in these cells. So we've proved that we could drive these cardiomyocytes to proliferate. However, this isn't a therapy, and this isn't driving cardiac repair. It's really just proliferating cardiomyocytes in the heart. So we decided to do another experiment where we overexpressed MYC and cyclin T1, again genetically, in a localized way, following giving this was in mice, giving them a heart attack. And what we could see is that in these damaged regions, we were proliferating cardiomyocytes. Again, the green cells, or the green is a marker of proliferation. And so we could drive these cells to proliferate. And if we look at actually the functional benefits, so this is a readout of heart function. So it is the amount of heart leaving every time the heart beats. And we can see in our mixed cyclin T1 overexpressing samples, their hearts had increased function. So we regenerated the hearts. And if you looked at the scar size, we had a significant decrease in scar size. So we remuscularized these hearts. But this wasn't a therapy. We did this in a genetic modification way. So we've been trying to develop a new therapy. And what we've done is we've selected mRNA therapy. So probably everybody in this room has already had an mRNA therapy, and that's your COVID vaccine. And so this is a way of overexpressing certain proteins. And so we've developed this therapy where we overexpress MYC and cyclin T1 by direct injection. And what we can see when we do this is that we see our proliferative markers in cardiomyocytes, and we get increased benefit again. So we're really excited by these results. We're trying to translate them further. There's still so much work to do. This benefit isn't quite as good as we saw in the genetic model, so we're trying to make it better. We're trying to explore it in other models. And this is where we come to John's talk. It would be really useful. I mean, this is all not done in human tissues and in human hearts, but to be able to confirm that our models work in a human setting, in a diseased organ that we want to put it into eventually, would just be phenomenal for our research. So I'll pass you over to John now.